Okay, welcome, ladies and gentlemen, to session 2 of the NBCEC's 2018 Brown Bagger. This second session features a couple of well-known speakers from 2 land-grant universities here in the States. We'll get to them in just a second. I want to point you to our NBCEC NBCEC website. As we progress here through the series, we're recording each session, and we'll post those under the For Professionals tab here, kind of in the middle. There's a page dedicated to the archives of the Brown Bagger series, and we'll have those YouTube-based recordings posted up for you to go back or share with colleagues as needed. Okay, I'm again Bob Weber. I'm a professor here at K-State. Work as one of the co-coordinators of the educational programming for the NBCEC, along with Dr. Matt Spangler at the University of Nebraska and Dr. Dare Bullock at the University of Kentucky. But I'll be flipping the levers here today, and again appreciate everyone joining us for this session 2. Session 2's title is Novel Trait Development, and we'll have 2 speakers this week. Our first speaker is Dr. Scott Speidel. Scott is on faculty at Colorado State University, and he joined the faculty there in July of 2014 as assistant professor of beef cattle breeding and genetics. Scott received his undergraduate degree in animal sciences from California State University, Fresno, where he spent most of his time there working in swine and dairy programs. His master's in animal science, specializing in dairy molecular genetics, was earned in 2001 at the University of Arizona under the direction of Dr. Sue Denice. Scott moved to Fort Collins during the summer of 2001 and began working as a research associate performing genetic evaluation at Colorado State University Center for Genetic Evaluation of Livestock. That's where I first got to know Scott. But Scott received his PhD in breeding and genetics in 2011 under the direction of Dr. Mark Enns, and the topic of his dissertation was the development of a random regression model for the prediction of days to finish in beef cattle. His research interests include identifying alternative methods for improving currently existing genetic evaluations for reproduction, and cow herd efficiency traits. So Scott is going to speak today about a novel trait that he's been working on with his team there at CSU on the genetic evaluation for PAP, or pulmonary arterial pressure. So Scott will be our first speaker. Our second speaker will be Dr. Megan Rolf. Megan's going to speak on water intake, testing period, and animal variation, some work that she's conducted at Oklahoma State and here at K-State. But Dr. Rolf's a faculty member here in our Department of Animal Sciences and Industry at K-State. Megan was raised on a cow-calf operation in East Central Kansas and has been involved with livestock her entire life. She received a bachelor's degree in animal science at K-State, a master's degree in animal science at the University of Missouri-Columbia. Actually, Megan was my second graduate student there. I'm pretty proud of Megan. She also earned her PhD in genetics at Mizzou under direction of Dr. Jerry Taylor, and her research there focused on implementation of genomic evaluations in crossbred beef cattle. Her first job out was at Oklahoma State University, where she spent 4 years on the faculty as state extension beef specialist. She joined the faculty here at K-State in 2016 as an assistant professor of animal breeding, and has a research appointment of 60% and 40% teaching appointment. She currently teaches genetics and maintains an active research program in the use of genomics for genetic improvement in livestock. So we're pleased to have both of you join us today. Scott, I'll unshare here, and if you'll pick it up, that'll be great. If I can remember how to stop share. There we go.
And is that, you hear me okay?
Yep, I can hear you loud and clear, Scott.
All right.
Okay.
All right. Does that look right?
Yep, we got slides. You're good to go, man.
Okay, perfect. Well, I appreciate everybody taking time to sit in with me today. Matt and Bob asked me to talk about genetic evaluation of pulmonary arterial pressure So the kind of the method I wanted to take today is, this is a prediction that's been around since, I believe about 1992 were the first estimates of heritability. And I want to talk about things like how it's progressed, particularly from a within herd evaluation, which is the way it started, to what we're beginning to see more trends toward breed-wide evaluations for pulmonary arterial pressure. I just want to take a real quick minute to thank some people. I mean, this is a piece of a much larger effort, a significant effort that we are at Colorado State University have been putting toward understanding pulmonary arterial pressure, and particularly its relationship with high altitude disease. I got the faculty in breeding and genetics that are currently putting a significant amount of effort, besides myself, is Mark Enns, Drs. Milt Thomas, and Dr. Stephen Coleman. I've got a bunch of grad students listed here. Not all of them are currently, presently grad students. Some have gone on and graduated and found jobs, but I'm presenting information. All these students have contributed to the information that I am going to present here today. And then we've got, I want to be sure to acknowledge our colleagues over in the School of Veterinary Medicine, Tim Holt and Frank Gehry, and Greta Krafser, spending a lot of time understanding the physiology and pathology associated with high altitude disease and its relationship with pulmonary hypertension. We are collaborating with Kurt Stenmark and Dale Brown at the UC Anschutz Medical Center down in Denver, and some others, Becca Cockrum, Angela Canovas, Juan Medrano, and Joe Neary, have all contributed significantly to this effort. When we think about EPD development, when you develop a novel trait, since the topic is novel trait evaluation, the first thing that we need to consider is really, is there sufficient data for the trait of interest, right? Because I mean, we begin to collect data, and until we get a significant number of observations, and that number is going to be dependent upon the ultimate heritability of the trait. But we need to to fully understand how these traits relate to one another that are currently being considered or selected upon. We need to gather enough observations, and really until recently, there's been a significant push to sort of compile resources and garner the support for more widespread or wide-scale genetic evaluations. As I mentioned earlier, we've at CSU have been doing within herd evaluations for pulmonary arterial pressure since the early 1990s. And that still continues, but we are starting to see a lot more interest, particularly from breeds that want to understand, to want to provide these types of selection tools for their clientele. So, as we begin to collect this data, we need to estimate heritability and genetic correlations. So understanding the genetic mechanisms of the trait and its relationships with other economically relevant traits as appropriate. For any sort of polygenic trait, continuously distributed polygenic trait like PAP, it's really kind of easy to calculate the EPD, particularly on a within herd basis, right? So, if something's moderately heritable, it's not real challenging to do those types of predictions. However, when you begin to transfer from within herd to breed-wide predictions, there's a lot of things that we need to consider. And in particular, I'm going to talk about later on things like contemporary grouping, things like elevation, and particular for PAP anyways, things like elevation, and then really sort of reaching out and teaching or educating producers or ultimate users of these technologies on how they can best select individuals for use in their populations. So just a real brief background. I have a couple of slides here. I just wanted to say that, you know, pulmonary hypertension is really the cause of what we call high altitude disease or brisket disease, okay? So pulmonary hypertension is similar to high blood pressure, right? But it's developed due to really, its cause is due to a low oxygen environment. So as we increase in elevation, these cattle are housed in a lower oxygen environment. Okay, so this leads to what we call vascular remodeling. Okay, and then ultimately, because of this, the cardiopulmonary system has to work harder, and this leads to what is a right ventricular hypertrophy, which we have essentially an enlarged heart, and then edema results, and this is called brisket disease because this edema collects down in the brisket disease, as you see in this picture of this Hereford female right here on the slide. Okay, high altitude disease is prevalent in the mountainous regions of the western United States. Calf loss can be up to 3 to 5% of a calf crop or higher, and you know, some initial estimates have shown that this is about a $60 million annual loss to the beef industry. So we at CSU have being where we are located, have kind of taken the charge to study this. Okay. Historically, pulmonary arterial pressure is the indicator for this pulmonary hypertension, right? So this is like, we're measuring the blood pressure of the individual, just like you would if you went to the doctor and you go in and have your blood pressure measured. It's a fairly invasive process that it requires a trained veterinarian to really sort of pass a catheter down through the jugular vein in the neck, and then this catheter goes all the way down through pass through the right atrium of the heart, into the right ventricle, and then ultimately into the pulmonary artery, where they are able to measure a pressure. Okay, this phenotypic pulmonary arterial pressure, Dr. Holt, who is kind of the leader from the leader of the veterinarians of the training new veterinarians for adopting this technique or training in this measurement technique. He's developed in a paper he published in 2007. He's developed a sort of a risk guideline. So from a phenotypic standpoint, anywhere less than 41 millimeters of mercury of pressure was considered to be low risk at developing high altitude disease. If you had individuals who had pressures from 41 to 48, were deemed to be acceptable for residing at high altitude, but sort of dependent upon current situations. You may want to use caution with using these for breeding purposes. And then greater than this 48 millimeters of mercury, they were considered to be high risk at developing high altitude disease. Okay, so this PAP is used as an indicator for high altitude disease. So I've compiled a lot, on the next two slides, I've compiled a lot of different heritabilities and genetic correlations and of PAP from different studies. And these are pretty much all of the studies that I am currently aware of that have looked at these on a sort of a breed-wide basis. Starting all the way up with the earliest ends 1992 estimate of heritability of 0.46. And then as we move down through this table, we get a change in the populations. All these heritabilities have been estimated mostly on Angus animal, purebred Angus animals. Some are registered animals. Some of these populations have been research populations. Some of these have, we do have one multi-breed sort of a data set that we are currently estimating breeding values for, that contains 7 plus 7, more than 7 breeds, ranging everywhere from Charolais to Angus to Gelbvieh and Red Angus and a lot of Simmental. Okay. One of the things that we see in these heritability estimates, so as we move downward in this table, we're moving chronologically in order of these publication dates. And we see heritability estimates ranging, anywhere from 0.25 to 0.34. 0.46 is higher. And this weaning, I threw this in there just because it's a different trait. But typically all of our pulmonary arterial pressures are evaluated at a year of age. So this was some research that was conducted by a PhD student here that was here at CSU. She graduated back in 2013. And to put all these heritabilities in perspective, I did a quick summary on some of the works out of the Coots papers. So if we think of traits like weaning weight, yearling weight, carcass weight, of which we've made substantial gains in over the past couple of decades, heritabilities are very similar. We're talking a moderately heritable trait, and this trait is what we call highly polygenic. And I don't have a slide in here representing that, but if we think about these in terms of the amount of genetic variability that are, and the genetic variability that are explaining differences in phenotype, we can make genetic progress, or at least if anything else, we can select against those high or those risk animals, right? So we can identify the animals that would be adapted to producing progeny at elevation. So the next thing I've got here is presenting more genetic correlations between a lot of these different economically relevant traits and PAP. And again, as we move across the table, so as we move from left to right in the table here, that we're chronologically representing genetic correlations from different studies. This Shirley et al. In 2008 evaluated weaning PAP. So those are a little bit different. That's a little bit different trait than what we would see at yearling. But the last three, we are looking at very similar data sets, kind of with the exception of the Pauling, which has the data presented in the Zeng and Crawford, but this one is a compilation of sort of vastly different background type data sets. So what I mean is that it's a compiled database of 3 different data sources, which we use to do a lot of evaluation of elevation and genetic correlations with production traits. Some of the things to point out here, one of the things that we should notice is PAP's genetic correlation with many of our traits is fairly low, which, if you're thinking about this from a production standpoint, could be a good or a bad thing. I mean, I think it's a good thing if we are looking at, trying to use this to increase adaptability to high elevation, but it could be a bad thing if you're trying to study PAP and its relationship with pulmonary hypertension, because it's not really genetically related to a lot of our other production traits. There's some sort of nuances with this Pauling. These are, I wouldn't put too much weight on these estimates here, specifically this 0.56. I think those are artificially inflated due to some pedigree structures we have with dams, and I think that's kind of an artificial inflation on just this particular data set. But one of the things of interest that we're seeing here is the genetic correlation between PAP and ribeye area is 0.24. And we're seeing some similar trends, at least on a phenotypic level, between PAP and carcass weight. So as these animals are becoming larger, larger body size, particularly from their carcass characteristics, we're beginning to see increases in pulmonary arterial pressure as well. So there seems to be something going on here with these carcass traits that is warranting, I think, further investigation on our part, anyways. So that sort of wraps up the genetic aspects of PAP. You know, the heritabilities and correlations. It's a moderately heritable trait, and you know, genetic correlations with production traits are fairly low. I'm going to spend the rest of the time talking about the non-genetic influences on PAP. And if I presented some of this material at the Beef Improvement Federation meetings here in Colorado this past June. So if any of you were there and saw my discussion, you would see some of these slides, and I've added in some things as well. But you know, as we begin to transition from a within herd evaluation to I guess what you would call a breed-wide evaluation, we need to be able to account for differences in things like age and breed and contemporary group and elevation. You know, as you begin to compile observations or data from vastly different sources or vastly different locations, a lot of these things begin to make sense or begin to need to be accounted for when we do genetic evaluations. So I'm going to talk about a few of these specifically. When we think about age of PAP measurements, according to Tim Holt, he says 18 months is really the ideal time to measure, but that's not a very good time to fit into production or management scenarios, right? So we, you know, typically PAPs, you know, culling decisions are made, you know, weaning or at yearling at least. There are not a whole lot of decisions here in the Western United States, particularly at high elevation, that are made beyond a year of age. So our yearling PAPs are really kind of the key decider there on that's kind of how we settled on that yearling PAP. We do know there are breed differences, and I'm going to present some data that talks about breed differences, but body condition and fat thickness, at least phenotypically, influences. So heavier conditioned animals will have higher pressures. Contemporary group, as we begin to move to a breed-wide evaluation, proper contemporary grouping is extremely important because that has a huge influence on our resulting predictions. I'm going to spend a fair amount of time talking about elevation differences because as we begin to collect PAPs at different elevations, as you move farther apart in elevation, the usefulness of predicting high elevation decreases. Just because as you increase in elevation, PAP tends to increase by 1 to 1.5 millimeters of mercury for every 1,000 feet. And if you listen to Tim Holt's discussion or his talk about these individuals, he says that 33% of the individuals that he tests will actually increase more than that. So as you increase elevation, PAP is increasing fairly rapidly. And then some other things over here on the left: parasite load, ration, the percent concentrate, whether or not ionophores are being fed. Any sort of lung damage is going to contribute to higher PAP measurements. Sex of the individual, so males, females versus steers, so there's differences there. Obviously, technician, this is similar to looking at ultrasound. If you measure carcass ultrasound for carcass characteristics, technician plays a role in differences in PAP, and then again weather. Actually, temperature, colder temperatures result in increased PAP measurements. So if we think about age real quick, and I'm going to talk about the ones that I think that are most relevant to genetic evaluation. Think about age. We do know that as animals age, their PAP increases. So this is a study by Joe Neary that was published in 2015 in the journal Animal Science. And he evaluated or repeatedly PAP'd a group of steers and followed them all the way from about 100 days of age out to right before harvest. And, one of the things that he found was PAP score increases with day of age. So if we think about a change in 100 days, you would expect animals that are 100 days older to have a 3.1 millimeter of mercury increase in PAP just due to animals being older, okay? So when we begin to think about evaluating this, that's something that needs to be accounted for, and whether or not, this gets adjusted for age, or whether we sort of evaluate age in the model, that's one consideration that needs to happen when we group a lot data from a lot of different sources together. I've got a table of breed differences presented here. Okay, the breed differences are really sort of an indicator of this is. Don't look at this table and come out and jump out and say, well, this can't be true. I threw this in here because we collected some data from the Four Corners bull test that was done on the San Juan Basin Research Center. And so these are groups of unrelated bulls that came in to the bull test, of which were PAP'd. And we just did some simple means and least square sorts of regression analyses to look at the relationships between breed and PAP. See, these are bulls that have vastly different genetic backgrounds. Some have not been selected for high altitude. And the one reason I did throw this in here is just because I wanted to show that there are differences amongst breeds. So as we begin to look at multi-breed data, breed is something that we need to account for, or from a genetic standpoint anyhow. So contemporary grouping these is really important. And it's probably one of the more important things that we need to consider when doing a breed-wide evaluation. We're evaluating PAP measurement, so we need some type of way to group individuals together all the way up to yearling. We need PAP date, particularly elevation is important. Disease status, PAP technician. These are all sorts of the things that go into define contemporary grouping, to contemporary group these animals on a larger basis. Probably the most important contemporary group plays a huge influence on the resulting predictions. And this is particularly true if ranch is accounting for differences in elevation of PAP. And there's some things that I'm going to talk about here in the next couple slides that we're going to talk about different ways to model elevation differences with PAP. But contemporary group, proper contemporary grouping, particularly if animals have been have any sort of respiratory damage, so lung damage from disease, we need to think about grouping these individuals separately, and that's the types of information that needs to be collected when we group these animals. So elevation is probably the second most important, just from the standpoint of are all PAP measurements, because PAP is a function of the amount of oxygen. It's a function of the cardiopulmonary system having to work harder because of lack of available oxygen in the environment. So as we increase in elevation, animals at higher elevations have less oxygen in their environment, so their system and their genetics have to work harder. So we need to what we're beginning to see is there is some sort of there is a G by E, or genetics by environment interaction, in with regards to elevation with PAP. So PAP at lower elevation is not the same trait as it is at higher elevation. And what we've done is we've had a grad student who has attempted to look at, you know, how can we classify this high elevation PAP, high elevation from moderate elevation. So she's essentially, what she's done is she's plotted elevation solutions for different groups of cattle. Okay, so here we've got the elevation solution, and then on the x-axis we have an increase in elevation. So what we've found is as you begin to increase in elevation, we begin to see contemporary group solutions, or groups of individual, have higher average PAP scores. Okay, so we've sort of drawn a line in the sand here, and as this begins to increase, or these mean PAP, these PAP means begin to increase, we are classifying this as high elevation, so above 5,250 feet, and then anything below 5,250 feet, we are deeming to have come from a what we call a moderate elevation. Okay. So then what we've done, and what she's done, is we've looked at the genetic relationship between these, this demarcation. Okay, so we've actually done this a couple of times. The Pauling manuscript, which was published in 2018, these are the heritabilities on the diagonal and the genetic correlation above the diagonal. And then we had another student who's presented a different data set, that these individuals, or sires, that had progeny PAP'd at two separate elevations, above and below 5,500 feet in elevation. These are very close. The demarcation here is fairly close, but the things that we're seeing is PAP at high elevation has a higher heritability. So meaning that as animals are increase in elevation, the environment is causing them to express more of their genetic differences. Or we are able to observe a greater amount of the genetic variability associated with pulmonary arterial pressure than we are at low elevation. Okay, and then some of the stuff that we're seeing genetic correlations between high and moderate elevation between right at about 0.8, 0.80, and that's you know, very similar to what we would see for a trait like weaning weight and yearling weight, right? So and some of the additional work, which I don't have presented here because I was short on time, is as these observations are recorded farther apart from one another, these correlations tend to fall off fairly rapidly. So in summary, kind of a PAP EPD summary, there is a significant genetic contribution to pulmonary arterial pressure. In the last round of EPDs that we ran, the PAP EPD range from -6.9 to just over 26 millimeters of mercury. We found that low elevation measures are strongly and not perfectly related to the high elevation measures. And so most likely how this information is going to be used is in a multiple trait analysis. So similar to what we would see from a carcass and ultrasound type scenario, we can use the PAPs recorded or measured at moderate elevations as an indicator of PAP at high elevation. We're spending a lot of time talking about how do we recommend we use these new predictions, just because the association-wide EPDs for PAP are soon to be released by the American Angus Association. And, you know, so some decisions need to be made on how we use sires. So, you know, historically what we've done, and just because of the nature of the beast, you know, people are comfortable looking at phenotypic PAP, but when we look at breeding values, we need to look at breeding values for, selecting those that are going to essentially produce progeny, right? So there's two different paths a sire can take, right? So the sires, you have the sires that reside at elevation, and then you have the sires that either through AI or some other fashion, some other technology will produce progeny at elevation, but not necessarily live at elevation, right? Or be housed at elevation. Okay, so our recommendations for that, just considering what we know about what constitutes a phenotype, right? So it's a contemporary group mean plus a breeding value plus some environmental effects. And we know that environmental influences on phenotype are not passed on to offspring, okay? But those environment play an important component to the individual's own phenotype, right? And that's the phenotype that individual has as an indicator of their ability to live and survive, or they're an indicator of their resistance to developing high altitude disease, okay? So if a sire is going to be housed at elevation, we need to look at their phenotypic observation in order to determine whether or not that's appropriate for their situation, okay? But for their selection to become parents, we should be looking at their genetic prediction. And part of that is because of, if we think about EPDs are being the prediction of the transmittable portion of phenotype from parent to progeny, right? And our modeling has a significant effort is made to reduce the environmental variability, to remove that noise from our predictions. These EPD will rank the individual animals according to their value as a parent, right? So this is so the EPDs need to be used for the prediction or the selection to become parents. So if we are considering an individual to be housed at elevation and we are considering a possible sire, one, we need to look at their actual phenotypic PAP observation to determine whether or not they're suitable for elevation. But in order to become a parent or use as a breeding individual, we need to consider make that sort on the basis of their EPD. And some of the discussion that we've had with some producers and Tim Holt, and we sat together in a room and have looked at these different EPDs and asking, what's a good EPD, what's a bad EPD. So PAP, I mean technically PAP lower is better, right? So we want lower PAP individuals. So kind of our general recommendations, at elevation, positive EPD, you should really use caution when using, just because, positive EPD tends to, we look at the individuals that have positive EPD who produce progeny at elevation, their progeny tend to have problems, okay? 0 or below, if an EPD of 0 or a negative EPD is good to use. But if you truly have problems in the herd, we need to have EPDs less than 0.7. Okay, if you have significant problems, that number may need to be lower, right? So this is all these are just sort of general recommendations. But individual circumstances will vary greatly here. So I don't know where I am on time. I probably went long, but
just a little, excuse me, just a little bit, Scott. But that's all right. And I know you've got to get to another engagement here shortly. So if there's a question or two. If you've got a sec, we'll take those now from the audience. We appreciate that. Was a really excellent presentation and discussion on PAP, and I know as a trait getting a lot more attention across the country due to the work that you guys have been undertaking here at CSU, and we really appreciate that that update. So, any questions for Scott? Scott, I'll throw one at you.
I was going to say, it sounds like my my undergraduate animal breeding class.
Crickets, right? Crickets.
They
Yeah.
either get it or they're lost.
Yeah, right. You talked a little bit about sort of, and I know your team has a paper out in JAS, I think the Culbertson paper, and looks at sort of that threshold of less than 5,000 or 5,200 feet.
Mm-hmm.
And higher elevation. It looks like maybe it's maybe a little bit nonlinear in that, as we get higher elevation, it's not necessarily just a linear increase in PAP, but maybe a little curvature to it. Do we get some additional benefit from evaluating animals at higher elevation if we can? More spread in those PAP scores, I suppose.
Yeah, I would high elevation is and it's the struggle with it is it's all dependent upon who the elevation at which the individual is going to be used, right? So we've got some range from 5,200 feet to almost 9,500 feet, and I know of some instances where animals are being housed even higher than that. So it's kind of for me it's kind of this is the start of a journey here, but you are correct. The closer we are to the intended use, the better they are a predictor of that PAP. So meaning if we are intending to use these individuals at 9,500 feet in elevation, a 7,500 foot PAP is a much better predictor of that than a 5,200 foot elevation PAP, if that makes sense.
Yep, absolutely.
And then so some of the stuff that I've been working on as we move forward, we've got some, I've been looking at some different types of analyses that allow me to model those, that relationship. That we are seeing more of a nonlinear relationship, and that difference in genetic correlation varies drastically depending upon the endpoint that we need to be at. So,
cool.
So yeah.
Good. One other question through the site here is, is PAP being evaluated anywhere else in the world? So I'm thinking about maybe parts of Europe that have maybe a big Simmental influence, like Switzerland, for example. Any other work going on around the world that you're aware of?
Not that I am aware of. I do know that PAP has been, from a from an evaluation standpoint, I am unaware. I do know that it has been measured in many other parts of the world. Tim Holt has traveled all over the world. He's traveled to Europe, Asia, Africa, and he has measured India, and he has measured PAP on these cattle. So I'm suspecting that there is some interest, but as far as a research standpoint and publication standpoint, I am unaware of anything else that's been done.
Excellent. Last question for you here. Question about what are the heterotic effects on PAP?
So heterosis, we do have a heterosis estimate, and I didn't put it in there because it's almost zero. I think the estimate is, if you go from the estimate for heterosis is a negative 0.04 millimeter of mercury. So as you go from the purebred to the F1 cross, you can expect a decrease in PAP pressure of 0.04 of a millimeter of mercury. So, which really isn't kind of that surprising. I mean, it's moderately to highly heritable, but I think that as we gather more data, there may be and the types of animals that we're looking at the heterosis, if we get more vastly different biological types of individuals, that might be that estimate might change.
Excellent. Great. Well, Scott, thank you again. We'll switch to Megan here in a sec, but again, really appreciate your effort joining us today and your valuable presentation on PAP. Thanks, Scott.
All right. Well, thank you. You have that back?
I got it back.
Okay,
Megan,
perfect.
if you'll go ahead and pick it up, that'll be great.
All right. Well, thank you, Bob.
Yep. Take care, Scott. Thanks.
Yep.
Okay, do you see slides, Bob?
I do, Megan.
Perfect.
Great, thanks, Megan. So our next speaker is Dr. Megan Rolf. Megan's assistant professor here at K-State and been doing a lot of work through a USDA-funded grant on water intake. And so we've asked Megan to give us a bit of an overview and update on that project and tell us what she's learned about water intake as a phenotype and how to measure it, and a little bit about what she sees in terms of variation of animals under testing protocols. So, Megan, the floor is yours, and thank you for joining us. And I know we ran a little bit over with Scott, so feel free to take your time. We've got the room till 1:30, and I know several attendees may have to step away, but we'll have the recording up shortly. So if you want to come back and pick up the end of Megan's talk, you're sure welcome to do that. Megan, thank you.
Great, thanks, Bob. And thank you all for tuning in. I will try and stick to my time slot as much as I can so you can get out of here on time. But really pleased to have the opportunity to tell you a little bit today about some water intake work that we've been doing in beef cattle. And much like Scott's project, there's a lot of people involved in this. I'll be sure to acknowledge all of those at the end. So one of the common things that people will sort of ask when I mention that we're working on water intake is why would you care about that particular trait? And I think for me, there's a couple reasons why I think it's interesting. And one of those is, of course, that water is one of our 6 essential nutrients. So we know that water has a lot of involvement in various metabolic processes and things like that, and we obviously can't live without water. So I think it's interesting from that perspective. And the fact that it's been tied to various performance measures in the literature. The specific mechanisms for that tie are sometimes a little bit less well defined, and it hasn't really been explored a lot from a genetic standpoint, at least within livestock species. A little bit of work been done in model organisms, and a little bit of work's been done with very specific water conditions like high sulfur and things like that. But when I think about water, one of the things that sort of prompted the focus on that for this project was drought. And the fact that we do have water shortages in some areas. So a lot of areas of the country and a lot of areas of the country where we tend to have a lot of cattle are areas where we don't necessarily have large amounts of rainfall, and we may have issues related to water rights. And of course, whenever we don't have a lot of water, we have a lot of competition between other agricultural enterprises with municipalities, of course, wildlife, things like that. And all of those issues can be exacerbated whenever we have a drought. So for me, that's kind of why I think water intake is important. But depending on where you're at within the country, it may be something that you think about much more in some areas of the country than others that maybe have lots of rainfall. So if we take a look at the literature and what we actually know about water intake, we know that there are breed differences, and specifically that cattle with sort of tropically adapted germplasm or Bos indicus-based breeds tend to have lower water intake, particularly when temperature and humidity levels are higher. We'll see higher levels of intake in Bos taurus cattle. There's also a little bit of work that shows that cattle will voluntarily restrict their water intake when water quality is poor. So particularly high when we have high salt, and there's some evidence that high sulfur concentrations, anything that affects the palatability of the water will impact their water intake. And anytime we have a water restriction, there's a good chance that it will impact body temperature regulation of those animals and downstream levels of heat stress. And that can become important when we think about sort of our classical summer drought when temperatures may be high. Beyond that, if you take a look at the NRC, I believe this is true even in this latest version, that a lot of the information based on water intake in the NRC is based off a paper of Winchester and Morris that was done back in kind of the mid-1950s. So I would argue that regardless, we have a need to collect some more contemporary data and make sure that water intake hasn't changed substantially in the last 70 years. So there's quite a bit of information on water intake in the literature for cattle that have been reared in pens. So particularly feedlot cattle where we put a water meter on the line coming into that pen, and we can look at total intake of the pen per day and sort of extrapolate that out to those individual animals. And in those studies, what you see is that the summer intakes are typically around twice what the winter intakes are on average, and there's a few studies that would back that up. Also, some water intake prediction equations have been developed in some of those studies as well. But we now have the technology to collect a large number of water intake phenotypes on individual animals. So this has been done in both dairy and beef cattle. And you can see those first two studies on the slide there have quite a bit of range in those values. Those are for Holstein bulls, and then you see the biggest range in those lactating dairy cows, which I think probably makes some sense. And then the last study on there, Brew and others in 2011, was performed with 146 growing beef cattle, and I believe they were at kind of thermoneutral type conditions. And their mean water intake was about 30 liters per head per day, so pretty comparable to those pen studies. They also found that animals that were out of bulls that were tropically adapted, of tropically adapted breeds, drank less than the British and continental cattle that they had in their study. One thing that you don't see within the literature, or at least that I've not been able to find, are heritability estimates or any kind of genomic analyses of water intake in beef cattle or other livestock species. So there is some work out there in mice, and they've had very successful selection experiments selecting mice for either very low water intake or very high water intake. And I believe the heritability estimate in some of those is up till in the range of 0.6, so it's quite highly heritable in mice, it looks like. So we set out to get a heritability estimate and kind of look at the amount of variability that exists within water intake and its relationship to other economically important phenotypes. So we put together a group of crossbred steers. The data that you're going to see today is going to be on 579 of those steers. When we get all done with this project, we'll have data analyzed on about 800 steers, but today we're going to look at a little less than 600. Those steers were fed over 5 groups. So there are 3 summer groups in group 1, 3, and 4, and 2 winter groups for group 2 and group 5. We did collect complete feed intake as well as water intake. We also collected gain information and some other measures. All of that was done within an Incentech system. And these animals did have access to shade. They were fed a growing diet, and there are a couple of differences in the management of these groups. So just kind of as you're going through the data, try to keep in mind that groups 1 through 3 were managed using a slick bunk feed calling procedure, and 4 and 5 had ad libitum access to feed. But regardless of that, every group that we're going to talk about today had access to ad libitum water during the time period that I'm going to show you data. So all of the data we're going to look at today is from this baseline period where that blue bar is, where they had access to ad libitum water. These steers did also undergo a water restriction trial, which you see there with the yellow and the green bars. We're not going to, because of time, we're not going to be able to look at any of that data today. And we're still analyzing it, but we will look at some data from that baseline period. So if we take a look at just some simple statistics on that data, our mean intakes across those groups ranged from about 28 liters per head per day to just shy of 50 liters per head per day. And as you might expect, the intakes on the winter groups, which as a refresher are 2 and 5, are just a little bit lower than we see in those other summer groups. So if we look at the absolute minimums and maximums within and across each of these groups, that's in that second box there on the right, you can see that they range from a minimum intake of about 15 liters per head per day, all the way up to one of the summer groups, we had an animal that drank over 100 liters in one day. So the first thing that we wanted to do was to take a look at test duration. Because there are guidelines for collection of feed intake phenotypes as well as for average daily gain, we wanted to take a look at whether we could collect feed and water intake at the same time and potentially even within a decoupled test like we now do with feed intake, or now have the ability to do with feed intake. So that was sort of our first objective here. And to accomplish that, we generated average intakes in sort of 7-day increments. So the first 7 days, the first 14 days, the first 21 days of the test, all the way through to 70 days. And we did the same thing in reverse. So the last 7 days, the last 14, the last 21, etc. And then we looked at the correlations of all of those values. And we did that within group, as well as within those different feed calling protocols, as well as across all of the different groups. And that's what you see in this table here. And I'm not going to spend the time to go through this entire table, but we have a pretty detailed analysis of all of this published in the Journal of Animal Science, and I've got that paper citation up here on the top right. So if you have interest in looking at that data in more detail, I'd encourage you to check out the paper. But the bottom line is, as we used a threshold of 0.95 for our correlations, because I believe that was what was used to establish Beef Improvement Federation guidelines for feed intake. So we use the same sort of threshold, and it looks like, based on our data, that you can collect pretty accurate water intake phenotypes in about the same amount of time that you can collect feed intake phenotypes. So the good news about that is it looks like we can collect water intake and dry matter intake phenotypes simultaneously, even if we have a decoupled intake and gain test. So we also did some work with looking at environmental effects on water intake. We don't have time to go through all of that today, so I simply wanted to say that our R-squared values, when we include dry matter intake, midweights, and environmental parameters within the prediction, will top out at around 0.4. So there's a lot of individual animal variation that's not really tied, doesn't appear to really be tied very closely with these environmental parameters. So if you have interest in looking at that analysis, that's published in the Journal of Animal Science as well, and I'd encourage you to check that out. But I do want to spend the rest of the time I have today talking to you a little bit about the water intake phenotypes that we have and some of the preliminary genetic analyses that we've done with that data. So as I mentioned before, we have water intake and dry matter intake records that we collected through that InSimText system. We did calculate average daily gain from our 2-week body weights by doing a linear regression of body weights on time. We went ahead and calculated feed to gain ratio and water to gain ratios, as you can see on the screen there. And we generated residual feed intakes using average daily gain and metabolic midweight, as well as residual water intakes, which we generated using dry matter intake and metabolic midweight. And so those are the phenotypes we were looking at here. The first thing we wanted to do was plot out and look at the phenotypic relationships of some of these different traits with water intake. And probably the one that you're going to be the most interested in on this screen is the one on the top left, and that is the relationship between water intake and dry matter intake. And here we had an R-squared value of about 0.14. And that was the highest R-squared value that we got with any of the other feed intake related traits. So water intake versus RFI is in the center panel, and water intake with feed to gain is in the top right-hand panel. Now the two on the bottom left are the relationships between the different water traits, and you can see those R-squared values are considerably higher, which is what we would expect. One thing that was fairly interesting about this is that we didn't see a relationship between water intake and average daily gain in this particular data set. So overall, we didn't feel like there were a lot of really strong phenotypic relationships between water intake and the different feed intake traits. So dry matter intake, residual feed intake, feed to gain, and we didn't really see any relationship with average daily gain at all. We did see moderate relationships between the water-related traits, as I think you would expect. So we wanted to estimate genetic parameters for these different traits. So we did that with a genomic bluff analysis because we did not have pedigree data on these particular animals. We know it's an admixed population of crossbred steers, but beyond that, we didn't have any information on ancestry. So we did that with the Bluff F90 software package. And for us, H inverse was essentially G inverse because we didn't have any pedigree data for those animals. We fit all these traits in bivariate models, so the values that you're going to see coming up here are going to be averages over all of those different bivariate models. For fixed effects, we included group and then the feed management protocol. And then for covariates, we included start weight and then various breed composition percentages because we knew that it was an admixed population, but we didn't have any other way to account for that. So we did that using the genotypes. So we used genotypes from the GGPHD150K to generate the genomic relationship matrix within BluffF90, but we also utilized it to estimate breed composition using a multiple regression-based method. And Larry Keene was kind enough to provide allele frequency estimates, since this is an allele frequency based analysis. And those allele frequencies came from larger populations of purebred animals in a variety of different breeds. And so the results of that analysis are here on this screen. So what you can see is that we had a lot of Angus ancestry in those animals, which I don't think is surprising. Some of the groups had high percentages of Red Angus, Hereford, Limousin, and Simmental. But overall, probably the takeaway from this is that we had a lot of different breeds involved in this particular analysis. And I do want to point out that we, in the selection of animals, we didn't have pedigree, but we knew there were probably differences that we would note from animals of Brahmin ancestry or potentially those with dairy ancestry. So we did try and just visually select against those animals when picking animals and putting them into the study. So you can see we were moderately successful in doing that. So we have a fairly low level of dairy influence in all of the groups. And in the first 3 groups, we do have a little bit of Brahmin influence in there. We weren't able to get that completely out of there, but we did include that in the model. Didn't feel like we had big enough populations other than maybe an Angus to actually estimate breed effects. So what we did was we grouped them into biological types instead. So percentage of British, continental, Indicus, and dairy ancestry were values that we used within that analysis. So if we take a look at the results of that, I'm going to point out the heritabilities on the top right there. The arrows point to our water intake traits. The rest of those traits, there's plenty of heritability estimates out there in the literature. But for water intake, residual water intake, and water to gain ratio, all of them were just shy of a heritability of 0.4. So pretty moderately heritable across all of those different traits. We also have genetic correlations here on the bottom. I've put a box around the genetic correlations with water intake because I think those are sort of the most interesting values in the table here. And what you can see is that we had high genetic correlations between water intake and residual water intake and water to gain, which I think we expected. We have kind of moderate correlations with dry matter intake and residual feed intake, right around 0.33 to 0.34. And then really no genetic correlation between water intake and average daily gain. You'll notice the feed to gain over there on the right-hand side is one that would fit in that high genetic correlation box, but you'll notice there's some kind of funky things with the standard deviations on those. So I do want to point out that, one, this is an analysis with a fairly small number of records at just under 600. So you'll notice the standard deviations are high on some of these. But it is a work in progress, so we have a little bit of work to do over there on some of those. So you might take those over on the right-hand side there with a little grain of salt. We're still working on those. But feed and water intake efficiency traits appear to be moderately heritable, and we didn't see any really strong antagonistic genetic relationships between water intake and either dry matter intake, residual feed intake, or feed to gain ratio. Although the relationship with feed to gain is strong, but I think we still have some more work to do on that particular analysis. Didn't really see any relationship with average daily gain. One of the other things we wanted to look at was the relationship of water intake with other types of traits. So that's, I'm going to show you a little bit of data there. It looks like we have time to go over just a small amount of that. With steers, we obviously didn't have reproductive data, which I think would be quite interesting, but we did have data on carcass traits. So I do want to point out that those the carcass data was collected after that entire test period. So the water intakes from that 70-day baseline, but they did undergo a water restriction and then a short finishing period before that carcass data was collected. It was collected using cameras at the packing plant whenever possible. There was one location where we weren't able to do that, but that only affects a small number of animals, and we did go ahead and collect those with trained personnel. And final body weights were measured within 24 hours of harvest. So here's a table that shows where those animals actually went in terms of the plant and how many, and then the days on feed, which reflects the days in the finishing period for each of those animals. And you'll notice there's a lot of variation in that table, and the reason for that is that the cattle enrolled in this particular study were producer-owned cattle. So because of that, these animals were marketed and went to the plant in response to changing market conditions, and we didn't have a lot of control over the ability to send, really large groups and things like that if market conditions dictated other practices. Methods for the genetic parameter determination or estimation were identical to the previous study, except for with the fixed effects for any analysis that included water intake, they were exactly the same as before, group and feed management, but for the carcass traits, it was simply group. For any analysis that included water intake, we used start weight as a covariate. For anything that included a carcass trait, we used days on finishing ration as a covariate, as well as our percent of reproduction. So if we take a look again at the phenotypic relationships between these different traits, what you'll notice is most of these lines are pretty flat. And essentially, we didn't really see any strong phenotypic relationships here. So the phenotypic relationships between water intake and carcass traits were pretty uniformly weak, and our highest R-squared was 0.05. So heritability, average heritability estimate in this was very similar to before. It was just slightly higher at 0.42. But in the purple box, you'll see the genetic correlations between water intake and these various different carcass traits. And what you'll see again is that in a lot of cases, these standard deviations are high, again, because we have a small data set, but the largest genetic correlations numerically would be hot carcass weight, back fat, and then close behind, we would have yield grade and final body weight. Marbling was just a little bit lower than that at 0.17, and then ribeye area, we didn't really see much of a genetic correlation there. Again, standard deviations are high on these, so some of them do overlap 0 on a few of these. So the genetic correlations between water intake and the carcass traits were around 0 to slightly positive. So there are potentially a few slight antagonisms with hot carcass weight, marbling, and final body weight, but again, large standard deviations. It'll be interesting to look at these again when we have all of the animals and all of the phenotypes available for analysis, which hopefully will have genotypes back on those within the next month or so. Looking ahead to the future, we do have a lot of other data to analyze, and luckily we have quite a few graduate students going to work on that. The first is a continuation of analysis on and including the last 2 groups of animals, which will be about an additional 240 records, as well as really digging into that water restriction period and looking at some of the adaptability data. And Jessica Neal is a PhD student that just started with us in August that's going to be starting on some of that. Is already working on some of that, actually. We also have Devin Jacobs, who's a master's student who's been with us since January. She's going to be working on some of the RNA-seq data that we're going to generate on some of these steers. We also have Lena Bond, who is a master's student that just started with us in August, and she's going to be digging into some of the phenotypic data and looking at some of the feeding behavior information that we have in there, as well as doing some analysis of sequencing data as well. Much like with Scott's project, this is a very small part of a much larger whole. So there are some other analyses going on at Oklahoma State University, as well as analysis of the metagenomic sequencing data that we have on these cattle, and that's being led by Udea de Silva. So to kind of wrap this up, looks like our test link that we need for water intake is compatible with a decoupled feed intake and gain test. So provided that a facility would have the ability to measure that phenotype, we do have the potential to get 2 phenotypes for the price of 1, if you will. Water intake is moderately heritable according to the population and data that we have. Antagonistic relationships with feed efficiency related traits and carcass traits appear to be pretty low, and shouldn't seriously hinder selection efforts if this is a trait that we decide we want to pursue. Although the caveat with this particular trait, which is probably the caveat with all traits, is that incorporation of a trait like this into a selection index would probably be sort of the best way to go on something like this. I do think water intake may pose some sort of additional questions related to sort of the best way to do that. So the importance of the trait and the difference in the importance of trait within different geographic regions may require some more personalized tools. So maybe incorporation into a decision support system or ability to build a custom index or something like that. With that, I do have some acknowledgements I need to give. The first is that I want to acknowledge Bob Weber, Jenny Borman here at K-State, as well as Larry Keene at US Meat Animal Research Center. They've all been involved in the analysis of the data from this project and have been really helpful as we work through all of this data. There are a lot of people involved in the original grant, which you see in that second list there, that have been working on various aspects of this project. We also have current and former graduate students at Oklahoma State that have worked on this project. And I want to acknowledge USDA for their support of this project. And we did also receive a little bit of supplementary funding from the Texas Cattle Feeders Association. And last but absolutely not least, especially in this case, I want to recognize Cashley Alberg. So Cashley was a PhD student who finished her degree in May. And so now Dr. Alberg has graduated and she's in Nebraska at the moment. But all of what you saw today was analysis that Cashley did as a part of her dissertation. So I think she did a great job with this data set. And I wanted to make sure I give her ample credit for generating all of the analysis that you had a chance to see today. So with that, I'd be happy to answer any questions if we have any.
Great, thanks, Megan. That was outstanding, and yeah, good shout out there to Dr. Alberg. She did a great job while she was here at K-State working on that project, and I know she was deeply involved in that work, and that's great. Attendees, if you've got a question for Megan, you can post that two ways. Either the Q&A tab on your screen there, you can post a question there, or through the webinar chat box. Either one of those is fine. I've got both of them open now on screen. So sure, if you've got a question, go ahead and post that up. As you're doing that, I'll throw one to Megan. You hinted a little bit at sort of others collecting data, which made me think about water data collection and genetics work in other species as a measure of adaptability. And are you aware of any, sheep data in particular is one that comes to mind, maybe an area where folks might have done some drought or water availability tolerance stuff? What's out there?
Yeah, that's a great question. Within livestock, I know there's been a little bit of work, not necessarily in the US, but overseas that has looked at different sort of watering patterns and how that affects water intake. So like, as an example, with goats, if they have continuous access to water versus access a couple times a day versus access once a day, how that affects intake and performance of those animals. So there's a little bit of that out there. Langston University has also done a little bit of water intake work with sheep in particular. So that's one of the other things that Devin's working on analyzing. Got data on, I think, just shy of 200 sheep that would have a water intake data, water restriction data, as well as feed intake and feed restriction data. So she's working on the genomic analysis of that particular data set. But that's also quite a small data set as well. So I think one of the interesting things that will come in the future for some of the water intake work in beef cattle is some of these Incentech facilities have now been installed in quite a few other places that have the ability to collect water intake. So hopefully in the next couple of years, as people collect more phenotypes on water intake, we'll be able to do some kind of joint meta-analyses to not only verify that the test length calculations and analysis that we did is consistent across multiple locations, which I think would be a really good thing to do, But we can kind of combine data from multiple locations, and as always, more data is better on a lot of that. So I think as we generate more of those records and can combine those into analyses, we'll be able to do a more precise job of estimating those heritabilities as well as the genetic correlations in particular. So.
Yeah, interesting. What other questions do we have for Megan? We've got a little bit of time yet that we can field those. Again, if you've got a question, you can pop them out there in the Q&A or webinar chat boxes. Either one's fine.
I might be off the hook, Bob.
You might be. Well, I got another one. I came prepared. So, one of the things I think is really really interesting, and particularly the genomics part of this work, is potentially identifying some genes that are responsible, or regions that are responsible for differences in water intake and water use. As you look through the comparative literature, what genes kind of stand out, and what tissues kind of are responsible? Things that come to mind are maybe some kidney function, that kind of stuff. Certainly relationships to gut health, and maybe assimilation of water across the large intestine, those sort of things. What sticks out in the literature to you?
Yeah, one of the things I've looked at a little bit is if you go into some of the different biological pathway databases, there's some stuff on like osmoregulation within the kidney in there. And it appears that the aquaporin gene families are pretty involved in some of that. So I think that's a possibility. One of the things that we did that wasn't necessarily a funded piece of this particular project was we collected tissue samples on all of these animals that we could within the plant. So one of the things that Devin and Lena and Jessica will be working on is we're going to generate some RNA-seq data from those samples, particularly the kidneys and the livers. Mm-hmm. And we'll probably be able to answer that question once we get those analyzed.
Cool. Sounds great. We'll look forward to keeping an eye on that research as it progresses. Any other, this will be last call. I know we've gone just a tick over time here, but that's all right. We've got plenty. If somebody's got a question, we can lob that in. We'll make that plea one more time. But Megan, sure appreciate your time in preparing your slides and comments for us today. I think a really interesting body of research, and look forward to hearing more about it as the project progresses.
Thanks for having me.
I don't see anybody typing, so we'll dally off. If you'll unshare, I'll put up the promo for next week's program. Fantastic. Share. Here we go. Okay. For those of you attending, here's the promo for next week, session 3, October 24th, again, noon Central Time for the speaker start. That session is going to be hosted by my colleague, Dr. Matt Spangler at University of Nebraska. And the overall theme for that session is encouraging data collection and correct breed utilization. So a panel of speakers to start off with, and looking at ways to incentivize collection of additional phenotypes and genotypes, perhaps through either purebred breeders or commercial producers. And so the panel members for that session include Kelly Ritalik from Angus Genetics Inc., Shane Bedwell from American Hereford, and then Dr. Jackie Atkins and Leoma Wells from American Simmental. All of those groups have been engaging producers to gather some additional phenotypic data to go into genetic evaluation systems. And so we'll look forward to hearing their strategies and success stories as they've worked on those projects. And then our second speaker in the session will be Dr. Larry Keene from USDA Meat Animal Research Center. And Larry's going to talk about some of their recent research looking at breed differences for mature weight in the germplasm germplasm evaluation program there at Mark. So again, noon Central, October 24th, Dr. Spangler will lead the discussion, and we look forward to having you join us. Megan, thanks again for your help today, and we'll look forward to having everybody on board next week. Thanks so much.